Chapter 4 introduces us to the properties of carbon, isomers, and the major functional groups, and in Chapter 5 we'll take a look at the macromolecules in biology. All living organisms have the element carbon as one of their major components. It is a unique element in the sense that it is very versatile in the ways it can form bonds with other elements. It can form covalent bonds with up to 4 other atoms or molecules simultaneously. There is actually a whole branch of chemistry that is dedicated to the study of all the different compounds that can be formed with carbon, and that is organic chemistry. Then there are hydrocarbons, which we will meet in this chapter and in Chapter 5. Hydrocarbons are organic molecules that consist of only carbon and hydrogen. So a carbon backbone or carbon skeleton, and then hydrogen atoms attached to those carbons in numbers that depend on the bonds that are between the carbon atoms. In the next several slides, we're going to be taking a look at various isomers. Isomers are molecules that have the same chemical formula, but they end up having slightly different properties, or sometimes significantly different properties, because they have differences in the arrangement of their atoms. We can distinguish between 3 types of isomers: structural, geometric, and enantiomers. Structural isomers have differences in the arrangement of the carbon skeleton. So the carbon atoms, how they are connected to each other. In geometric isomers, we see differences in the organization of carbon atoms or of groups of other atoms across a double bond between 2 carbon atoms. Geometric isomers are also known as cis-trans isomers. And then finally, enantiomers are molecules that are mirror images of each other, and they are characterized by the presence of a chiral carbon atom. In other words, a carbon atom that is surrounded by 4 different chemical groups or 4 different elements. Butane and isobutane are 2 examples of a structural isomer. They each have the formula C4H10. So they have a carbon skeleton composed of 4 carbon atoms, and those 4 carbon atoms are surrounded by hydrogen atoms, and they all have single bonds, single covalent bonds. If we compare the 2 skeletons, we have butane here on the left and isobutane on the right. So the 4 carbons are here. Carbon number 1, 2, 3, and 4 for butane. And for isobutane, you have 1, 2, 3, and 4. When you compare them to each other, you can see already that they each have 4 carbon atoms. If you count the number of hydrogen atoms that surround the carbons, then you'll see that each has 10. I'm not going to write each of them down, but this is hydrogen number 1, 2, and so forth. So they have exactly the same number of both carbon atoms and hydrogen atoms. What is the difference? Well, you can see that the way the carbon atoms are arranged is different. In butane, the carbon atoms are connected to each other as a single line. So carbon, 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 carbon in a straight line. In isobutane, there is a branch. So there is the longest line of carbon atoms is 3, and then the carbon atom in the middle has another carbon atom branching out from it. And notice the total number of hydrogens is still the same between them, because each carbon atom has to have 4 covalent bonds. So let's say if we look at carbon atom number 2 of butane, it has 2 covalent bonds with other carbon atoms, carbon atom 1 and 3, and then 2 more with 2 hydrogen atoms. If we look at, say, carbon number 4 in isobutane, it is connected through a covalent bond to one other carbon atom, to number 2, and then 3 covalent bonds to hydrogen atoms. What is the difference between the two molecules? They're similar. They are both gases, but look at the boiling points. Butane has a boiling point of -1 degree Celsius. So above -1 degree Celsius, butane is a gas. Isobutane's boiling point is -12 degrees Celsius. So it is a gas above -12 degrees Celsius. So slightly different boiling points. Butane, you can find it often in gas canisters for portable stoves or for regular cooking gas. Isobutane is often used in lighter fuel. So they have slightly different properties, similar molecules, the same formula. So these are structural isomers, and the difference is in how the carbon atoms are connected to each other. In this little drawing of isobutane in the formula, this bond, the branching bond between the middle C and the branch, should be right on top of the C itself. It's moved over a little bit, but this is the position where it should be. Another example of structural isomers is pentane. Pentane comes in 3 structural isomers. N-pentane is the one on the left, and that is the unbranched form. It's known as normal pentane. And then you've got isopentane with 1 branch, and then neopentane it's like a globular molecule almost. So let's take a look at the differences. So in N-pentane, we've got all the carbons in a row, and you've got the hydrogen atoms distributed in such a way that each carbon atom has 4 covalent bonds. And the formula for all of them is C5H12. In isopentane, the longest row of carbon atoms has 4 carbon atoms. So you got 1, 2, 3, and 4 carbon atoms. And then each of them is surrounded by hydrogen atoms such that it will have 4 total covalent bonds. In neopentane, there is a central carbon atom here, and that central carbon atom is bound to carbon atoms on all 4 of its available slots. So all 4 bonds that this carbon atom can form are with other carbon atoms, and then those 4 carbon atoms each have 3 hydrogen atoms bonding to them. All 3 the same formula, the same composition, slightly different properties and shapes. A different type of isomer is the geometric or cis-trans isomer, and in those the differences are between atoms that are connected with a double bond. So we've got here 2 carbon atoms that are connected through a double bond, and then each of them have room for 2 more elements to bond with them. And in this case here, we see that on the first carbon atom, it has an X on the top and a hydrogen atom on the bottom. In the second carbon atom also has an X on the top and the hydrogen atom on the bottom. Compare that to its isomer. In the isomer, on one side you have the X on the bottom and the hydrogen on the top, while in the other one the X is on the top and the hydrogen atom is on the bottom. So those are the 2 types, the 2 versions. This one on the left is the cis isomer, and the other one here on the right we have the trans isomer. The terms cis and trans refer to the position of those substituent groups. Cis means on the same side. So in the cis molecules, the 2 X's are on the same side of the double bond, while on the trans molecule they are on opposite sides of the double bond. Here is an example with an actual molecule. So you've got a molecule called 1,2-dichloroethene, in which the substituent is a chlorine atom, here in green. In the trans isomer, the 2 chlorine atoms are on opposite sides of the double bond, and on the cis isomer they are on the same side of the double bond. So cis-trans isomers are also known as geometric isomers. The 2 molecules we looked at in the previous slide, 1,2-dichloroethene, that isomer pair is shown over here using models, plastic models, to make the molecules. The double bond is here, and then the 2 chlorines are represented by the green balls. So you see the cis isomer on top and the trans isomer in the bottom. And then these 2 are a different pair of isomers, and they are 1,2-methylethene, and the substituents are 2 methyl groups. So here is one, here's the other, and this is also the cis isomer because the 2 methyl groups are on the same side of this double bond. In the trans isomer, the 2 substituent groups are on the opposite side of the double bond. So they're basically diagonally from each other over here. Enantiomers are isomers that are very similar to each other. Of course, they have the same formula and composition. They're based on at least one carbon atom that is a chiral carbon. In other words, this carbon atom is attached to 4 groups that are different from each other. In this example, we see that this central carbon is attached to an amino group, and then also a carboxyl group, a hydrogen atom, and a methyl group. So those are 4 different groups. Notice that its isomer has the same 4 groups, but they are arranged in such a way that the 2 isomers are mirror images of each other. This kind of isomer is sometimes also referred to as an optical isomer, and they are to each other like the left hand is to the right hand. Notice that you cannot superimpose the 2 hands, the right hand on the left hand. If you have, say here, the left hand, you got the palm facing you and the thumb is pointing to the left. If you wanted to have the right hand with the thumb also pointing to the left, then you would have the back of the hand facing you. So it would never be exactly the same. You'd need to have 2 left hands or 2 right hands for them to be superimposable on each other. Just like the right hand and the left hand are not superimposable, enantiomeric molecules are also not superimposable on each other. On the illustration, here we're looking at a set of enantiomers, and we just have them facing, in this case, sort of in opposite direction, and the chiral carbon in them, I'm going to circle it, is this carbon here. So it is attached to a methyl group over here, a green atom. Let's say that that's a fluorine, an orange one, a chlorine atom, and a hydrogen atom. So 1, 2, 3, and then let's make the methyl group number 4. So that makes this central carbon here a chiral carbon. This other molecule here is its enantiomeric twin, but it is arranged in such a way that the 3 different ones, the fluorine, the chlorine, and the hydrogen, are on the left side as we see it, while in this molecule they're on the right side. On the second molecule they're on the left side. If we rotate this guy so that now it has the green and the orange atoms also facing its right side, that's what we did on this side here. Now you see that now on this one the green atom is on the back and the orange one is in the front, while in the other one the green one is in the front and the orange one is in the back. So no matter what you do, you cannot exactly superimpose them on each other. Either if both the green ones are pointing to the front and both the orange ones are to the back, like we see in the situation on the left side, then on one molecule they're on the left and on the other one they're on the right. If they're both on the same side, then one is in the front and the other one is in the back. To illustrate the concept that enantiomers are mirror images of each other, let's actually have our molecule with the chiral carbon atom look in a mirror. So this is the original molecule and we called it number 1, And when we put a mirror in front of it, the image of it, number 1 prime, is its mirror image. Of course, it's in the mirror, but it is also, if it would be in real life, it would be its enantiomer. And it has the same organization, but in the opposite direction. Okay, now we're going to take this a step further. Remember, when we say that two molecules are superimposable on each other, it means that you can superimpose all of their atoms. So we've got now the original molecule number 1 that we had before looking in the mirror, and it sees its reflection, which is 1 prime. But now we have sitting next to it its enantiomer, which is molecule number 2. And molecule number 2, if it is the enantiomer of molecule number 1, then molecule number 2 should be exactly superimposable on molecule 1 prime. We can't see that right now. What we need to do is that we need to turn molecule number 2 around so that you can see that all the atoms in molecule number 2 are in the same direction as those in molecule number 1 prime, but not molecule number 1. So now we rotated molecule number 2. And if we compare, we see that first molecule number 1 with number 2, number 1 and 2 are an enantiomeric pair, and you cannot superimpose 1 on 2. However you rotate 1 or 2, either the green and the orange are on opposite directions or they are on the opposite side of the molecule. But if you compare 1 prime with 2 now, you see that everything is the same, right? You have this molecule, this atom, and is the same as that, and then this one in the front is the same, and then you've got the orange one here on the right, and it's on the right on this one, and you've got the green one on the left, and you have the green there. So everything is the same between those two. The mirror image of number 1 is the same as the enantiomer of number 1. In chemistry, two molecules that are enantiomers are basically exactly equivalent to each other. When you make molecules in a chemical reaction, if they are enantiomers, you usually make 50% of one and 50% of the other because it's random which one is going to be made. Sometimes you can separate them from each other because the crystals, one may rotate light in the opposite direction as the other one. In biology, however, our enzymes, which are the molecules that catalyze our chemical reactions, the cellular chemical reactions, the biochemical reactions, they do have a preference for one of the two enantiomers. So they can only use one of them because the enzyme is not symmetrical. We have here an illustration of two molecules. One is called L-DOPA and its enantiomer is D-DOPA. The one that is active in the body, that is effective against Parkinson's disease, is only L-DOPA. So if you are given a medication that has a racemic mixture that is chemically synthesized and it has equal amounts of both of them, that means that you're only going to be able to use the 50% that is the L-DOPA part. I think the same happens with certain vitamins. Sometimes when you buy the vitamins, it is an equal mixture of both enantiomers. Only one of them will be usable by the body. A functional group is a small group of atoms that tend to function as a unit, and as a unit, they will impart certain characteristics to the larger molecule that they are attached to. For example, solubility in water, or solubility in fats or oils, or alkalinity. There are many different functional groups, but for Bio 210, we're going to be looking at this small group of 7 functional groups, and in the coming slides, we'll be taking a look at each of them. The hydroxyl functional group consists of an oxygen atom covalently bonded to a hydrogen atom, and it tends to occur in abundance in sugars, as we can see here on glucose, where you have several different hydroxyl groups in the one molecule of glucose. And it also can be found in alcohols. The hydroxyl functional group in general tends to make molecules more soluble in water. The carbonyl functional group consists of a carbon atom double bonded to an oxygen atom. The carbon atom has 2 more places for covalent bonds on it. So it can be located, the carbonyl group can be located either on the inside of a chain of molecules, or it can be at the end of the chain, and the resulting complete molecule is going to have a different name depending on whether the carbonyl group is at the end of the chain or on the inside, in one of the inside carbons of the chain. If it is on the inside carbon or on one of the inside carbons, then that molecule is called a ketone. And if the carbon that has the carbonyl, that is part of the carbonyl, is at the end of the chain, in other words, carbon number 1, then the molecule is called an aldehyde. So both aldehydes and ketones have a carbonyl group. Glucose and fructose are structural isomers of each other. In other words, they have the same chemical formula, but they have a different arrangement of the atoms within that formula. If you take a look at them, one of them is an aldehyde, the other one is a ketone. So we were saying in the previous slide that if the carbonyl group is located on the first carbon atom, that makes the molecule an aldehyde, and if it is located on one of the internal carbon atoms, that molecule is a ketone. So we see from this that glucose is the aldehyde and fructose is the ketone. The Benedict's reagent is based on copper sulfate that can react with the carbonyl groups in sugars. Specifically, it has to be a free carbonyl group that can react with this copper sulfate and reduce it to copper oxide. That produces a reddish precipitate. And so you can see here that both glucose and fructose test positive in the Benedict's reagent, but sucrose does not. When you have glucose in solution, it alternates between the ring form, which we see here, and the linear form over here. So it goes back and forth between the ring form and the linear form. The linear form is the one that has the free carbonyl group. So this is the one that can produce the positive result in the Benedict's test. When it is in the ring form, it would not be able to reduce the reagent in Benedict's. But as it goes back and forth, as soon as it goes to the linear form, it gets oxidized, and then it gives the precipitate. It reduces the Benedict's reagent. And this here is the intermediate shape when the carbon number 1 with the carbonyl group reacts with the oxygen here, this hydroxyl group on carbon number 5. We're looking at some plastic models of the 3 shapes of the glucose molecule. So we have the linear form here, the intermediate over here, and the circular form here. When you have them in the models like this, you can see that to go from the linear to the ring form is not that much of a stretch, because in the linear molecule, the molecules are already pretty close together. In other words, the way the linear molecule is drawn with a straight line like that is not how it looks in reality. In reality, it looks more like this. And then when carbon number 1 over here with the carbonyl group reacts with that hydroxyl group on carbon number 5, they come together, and now you have the ring form where there is no carbonyl anymore. So remember, in the ring form, glucose would not react with the Benedict's reagent. The carboxyl functional group is COOH. This symbol that we see here is not a minus sign. It just symbolizes the covalent bond with which the carbon atom is attached to the bigger molecule. We can find the carboxyl functional group both in fatty acids, such as this fatty acid, palmitic acid, that we see over here, and in amino acids. Carboxyl is really a composite functional group, a composite functional group, in that it consists of a combination of a carbonyl functional group and a hydroxyl functional group. But remember that functional groups work as a unit. So now this new unit is known as the carboxyl functional group. Carboxyls make molecules acidic by donating protons to the solution, and they do this by the ionization of the carboxyl group. Take a look over here to see how that happens. Over on the top we have the carboxyl group before it has ionized, and here there is a covalent group, a covalent bond between this oxygen and the hydrogen. Remember that in a covalent bond, 2 atoms share a pair of electrons. One of the electrons is coming from hydrogen and the other electron is coming from oxygen. During the ionization, the oxygen is going to take both of the electrons, so it has its own electron and the one that it stole from the hydrogen atom. So now you're going to have that the oxygen has acquired a negative charge, so it is here O minus, and it is part of this COO minus that is left of the carboxyl group, and then an H plus will be released into the medium. and an H plus is a hydrogen ion, and a hydrogen ion is nothing more than a proton. Remember, a hydrogen atom is a proton that has an electron orbiting it. The electron was taken by the oxygen, so what is left is the proton by itself. We see the carboxyl group here in both the unionized form, which is the upper model, and in its ionized form. The unionized form, you remember, is written COOH. And in the ionized form is COO minus, with this oxygen over here having a negative charge and the proton being positively charged. The phosphate group is the other functional group that can make molecules acidic. The phosphate group has a phosphorus atom in the middle, and this phosphorus atom is surrounded by 5 oxygen atoms. One of the oxygen atoms is connected to the phosphorus by a double covalent bond, and the other 3 are connected through single covalent bonds. Notice that the phosphate functional group includes 2 hydroxyl groups, OHs, but again, remember that the functional group is considered a unit, so the phosphate group includes all of these. Phosphate can donate 2 protons to a solution in a similar way as how we saw the ionization of carboxyl, where 1 proton can be donated. In the phosphate group, both here and here, you can have an ionization and 2 protons can be donated. So you can have a situation in which the group can be completely unionized, or one of the 2 OHs can have ionized, or both. We see underneath a picture of a nucleotide, and the phosphate group is shown over here, and you see that both of those hydroxyl groups that are part of it are ionized here, so it has 2 negative charges. A nucleotide is a nucleoside plus a phosphate group. So the nucleoside part is these 2, a sugar and a nitrogenous base, and then plus phosphate makes it a nucleotide. Where do we find phosphate groups? We find them in the nucleic acids in DNA and RNA. Phosphate groups are extremely important in energy transfer also, as part of the nucleotide ATP, and we're going to learn more about that when we get to chapter 8. Also notice that phosphorus atom in the middle is participating in 5 covalent bonds. The amino functional group, NH2, can make a molecule basic or alkaline by accepting a proton. When an amino group accepts a proton, it becomes NH3+. And that is its ionized form. we find amino groups in the molecules known as amino acids, which are the building blocks of proteins. Here we see the amino group on an amino acid. Here's another representation of an amino acid, which is important in this part of the chapter because it has two of those functional groups that we've been looking at. The amino acid has both an amino group and a carboxyl group. Then there is a hydrogen atom and a group called an R or radical group, and all of them are connected to a central carbon atom that is known as the alpha carbon. We see that same amino acid represented here in a model. So here is the carboxyl group, here is the amino, and the hydrogen atom, the alpha carbon, is over here, and then the green ball represents the R group, and the R group can be a lot of different things. That is what makes each amino acid unique and different from the other amino acids. The sulfhydryl functional group is found in the amino acid cysteine, which we can see over here. I guess I have to use a different color. Cysteine. So that's one copy of the amino acid cysteine, and two different cysteine amino acids in a protein can link to each other through their sulfhydryl groups, and that's what we're looking at over here, where the two sulfhydryls interact with each other and form a covalent bond, a disulfide bond that we see over here. And that is used in proteins sometimes to attach different parts of a protein to each other. We see that on the right side illustration. I'm following the shape of a polypeptide, and it has these two cysteine amino acids that are connected to each other through a disulfide bond, and that is a stable covalent bond that will help determine the permanent and active conformation of a protein. The sulfhydryl group is also known as a thiol or thiol group. This picture shows us some of the places where you can find sulfhydryl containing compounds. Let's start with the garlic. Everybody probably has eaten garlic, and you can almost smell the cut garlic in this illustration over here. And everybody knows that characteristic smell that we find in garlic. Sulfhydryl compounds are also responsible for the smell of skunk spray. This very handsome skunk that we see here produces certain sulfhydryl containing compounds in the spray when he gets disturbed, and it's a very strong odor and difficult to get out of your clothes if you ever get hit. And then finally, grapefruit has sulfhydryl containing compounds too. So you can see they all have in common the sulfhydryl groups, but the actual smells are very different depending on what other molecules they are attached to. In the case of the grapefruit, it is also that the concentration is much lower than in, say, garlic or in skunk spray. If you concentrate that grapefruit containing sulfhydryl compound, it is also quite strong. The methyl group consists of a carbon atom that is covalently bonded to 3 hydrogen atoms. We find methyl groups as a structural component of fatty acids. The last carbon atom in a fatty acid, the omega carbon, is part of a methyl group. Methylation of DNA is used to turn genes off or on sometimes also. So it can be used to control gene expression. So the methyl group is also very important. In general, it makes molecules hydrophobic or not soluble in water. Estradiol and testosterone are very closely related molecules, as you can see over here. They're both derived from cholesterol. Here is estradiol, and then underneath we can see testosterone. You can see there are a few differences where estradiol has this group, a hydroxyl group. Testosterone has a carbonyl group. And testosterone also has this group here, a methyl group attached. And those small differences are responsible for the fact that testosterone or testosterone derivatives are used to determine secondary sexual characteristics in males, such as the mane of this lion, while estradiol is used for determining secondary sexual characteristics in females. Chapter 5 introduces us to the 4 macromolecules: carbohydrates, lipids, proteins, and nucleic acids. We learned in an earlier chapter that the cell is the basic unit of all life, and that all living organisms are made of cells. And this is true. In this chapter, we're going to take a look at the large molecules that cells themselves, the structures inside cells, are made of. These large molecules are known as macromolecules, and the prefix macro just means large. There are 4 types of macromolecules, which are the lipids, the carbohydrates, the proteins, and the nucleic acids. In general, macromolecules have smaller subunits or building blocks that make up the larger molecules. Of the 4 types of macromolecules, 3 of them, the carbohydrates, the proteins, and the nucleic acids, have the tendency to form extremely large molecules that are composed of individual subunits of the macromolecule strung one after another. So a polymer can consist of thousands to millions of these subunits, forming extremely large molecules. In the case of lipids, their their structure is less ordered, maybe more amorphous than that of the other macromolecules. So in lipids, you have hydrocarbon chains and you may have aromatic rings, but they don't form these extremely large molecules that have lots of repeating units strung together and connected to each other, as we will see with the other three macromolecules. In all four macromolecules, there are reactions that need to be performed to make larger molecules of smaller subunits, and that applies even to the lipids. So when you make a larger molecule from smaller subunits, those reactions are called synthesis reactions. Other names are dehydration reactions and condensation reactions. In synthesis, dehydration, and condensation reactions, we're taking smaller pieces and putting them together, linking them together into larger units. We have an example over here that shows a short polymer, and as you can see, there are 3 subunits in that short polymer, and at one end there is a hydrogen atom, and at the other extreme there is a hydroxyl group. And here we have an unlinked monomer. So monomer is really small, the smallest subunit of that particular kind of macromolecule. And it also has, on one end, it has a hydroxyl group, and on the other end, a hydrogen atom. In a synthesis reaction, we're going to take the hydrogen atom from one side and the hydroxyl group from the other side, and we're going to extract those, and a water molecule gets formed. So a water molecule gets released, and now we have a larger molecule, a longer polymer. In this case, it has 4 subunits. Because you release a water molecule in synthesis reactions, they're also known as dehydration reactions, where you're losing water, and condensation reactions. A hydrolysis reaction is the opposite of a synthesis reaction. In a hydrolysis reaction, we're going to be breaking a larger molecule into smaller ones, and the procedure is also the opposite. Where we saw in the condensation, dehydration, synthesis reactions, you are going to extract water molecules from the subunits. In the hydrolysis reaction, you're going to be adding a water molecule to the subunits, and that is going to help break them up. And this is why it's called hydrolysis. Hydrolysis means to break with water. If we look at the example, we see now we have the 4 subunit molecule, and it's got the hydroxyl group on one end and a hydrogen atom on the other end. Remember, there's no, those have been taken out from in between. But now we take a water molecule, and that water molecule is going to be inserted between subunits 3 and 4 in such a way that the hydrogen atom is going to go to subunit number 3 and the hydroxyl group goes to subunit number 4, and this breaks up the 2 molecules. Hydrolysis tends to release energy, so energy is released when molecules are broken, and synthesis reactions in general tend to consume energy. The subunits of carbohydrates are the monosaccharides or simple sugars. The word carbohydrate itself means hydrate of carbon, and it indicates that you would have a carbon atom that is attached to or associated with a water molecule. That name is actually a misnomer. You don't have carbon atoms that are together with water molecules in that sense. What you do have are repeated units. Like you can see on this fructose molecule, I'm circling CHOH, and then another one here, CHOH, and so forth. You see lots of those. If you just look at the chemical composition, you have a carbon, you have an oxygen, and two hydrogens. So that's like a carbon and a water. So that's why they're called carbohydrates. Now let's take a look at some of the other characteristics of carbohydrates. We see that in this table there is a distinction made between aldoses and ketoses. In general, aldoses have a carbonyl group. In fact, all monosaccharides have a carbonyl group. As you can see, I'm circling these two carbonyl groups on glyceraldehyde and ribose. And in aldoses, that carbonyl group is on the first atom of the carbon skeleton. So that makes them aldehydes. In ketosis, the carbonyl group is on an interior carbon, as we see here with dihydroxyacetone. It's not on the first carbon. So that's the difference between aldoses and ketosis. All monosaccharides have a carbon atom skeleton. I'm circling the one from glyceraldehyde. Glyceraldehyde has a 3 carbon atom skeleton. Ribose has a 5 carbon atom skeleton. So they have that carbon atom skeleton, and then they have carbonyl group. Let's see, here's the carbonyl group on ribulose. And then there are lots of hydroxyl groups. That's the other important functional group on monosaccharides. Lots of hydroxyl groups, and each of them has 1 carbonyl group. If we look at the chemical formula, the general chemical formula, for example, of the hexose sugars, that shows it is C6H12O6, you can see how that is a multiple of the CH2O that I was mentioning to you earlier. So this is why they call them carbohydrates, because you've got that CH2O subunit repeated multiple times. But you just have to keep in mind, it is not, the water molecule is not there as a unit. It's as a hydroxyl group, and on the other side a hydrogen, and each of them is attached to a carbon atom. We saw in Chapter 4 that isomers are molecules that have the same chemical formula, but different orientation or different organization of the atoms with respect to each other. Glucose and galactose are an example of that. They have exactly the same formula, C6H12O6, but the functional groups are arranged slightly different. One of the hydroxyl groups is oriented in opposite direction in one with respect to the other. That gives them slightly different physical characteristics. One may be slightly sweeter than the other. They may have a slightly different melting point from each other. Monosaccharides are capable of forming polymers with each other, and the first step in doing that is to form a disaccharide. And a disaccharide is two units of monosaccharide that are joined together through dehydration synthesis and then forming a covalent bond between them. So we see here on the left side, let me switch my pencil for a second. Okay, on the left side we see that you have a glucose unit, and they are bringing to our attention this hydroxyl group. And then next to it we have a fructose molecule, and they're showing us this hydrogen atom. And those two in blue are going to react together, and they're going to be extracted from each of those molecules and form a water molecule. So this is one of the reasons it's called dehydration synthesis, because you're losing a water molecule when you make the bond. And then what's going to be coming instead of that is this linkage, a covalent bond between the two monosaccharides, and that is known as a glycosidic linkage in the case of carbohydrates. A polysaccharide is then many subunits, many monosaccharides that are joined with these glycosidic bonds through dehydration, synthesis, or condensation reactions. And we have different types of polysaccharides. Some are used as fuel molecules. In the case of plants, starch is a fuel polysaccharide. So we see that over here. We see a chloroplast, and we see starch accumulating inside the chloroplast. In the chloroplast, we will learn in chapter 10 the reactions of photosynthesis produce glucose and other carbohydrates, and that can accumulate as starch inside of the chloroplasts. In animal tissues, a fuel storage carbohydrate is glycogen. We accumulate glycogen in the liver and in the muscles. And then there are structural carbohydrates, and those include cellulose, and cellulose is a major component of wood in plants. It's actually a long carbohydrate. And chitin. Chitin is a carbohydrate that has some other groups added to it, so it has some substitutions, and it forms the major part of the exoskeleton of insects. In the picture we see a cicada coming out of its pupa, and this brown structure here is mostly chitin. There is also chitin in the insect that is emerging, and later that hardens and it becomes the regular protective structure for the insect. The dark granules that we see here are starch granules in a potato. So this is an individual potato cell, and then you have the starch granules inside of it. The reason they are dark is that the potato slice has been treated with iodine, which then reacts to give you that blue-black color. Cellulose is a structural polysaccharide that is one of the major components of wood in plants, one of the major components of the cell walls in each plant cell. It is also present in softer plants like grass. So we see this cow here enjoying some nice grass. Animals actually don't have the necessary enzymes in their own bodies to digest cellulose. They cannot break down the chemical bonds that connect the glucose units with each other. A cow has in its stomach, or one of its stomachs, it has these bacteria that help with the digestion of cellulose, and therefore a cow can eat grass and can derive nutrients and calories from that grass. When we eat broccoli or other plants that have lots of green and lots of cellulose in them, bacteria in our intestine, in our large intestine, are able to break down some of that cellulose as well. But in many cases, the cellulose passes through our body unchanged, and it is what composes the fiber in our diet. In a similar way that cows can digest cellulose thanks to their bacteria, termites digest cellulose with protozoa that are present in their guts. Without these protozoa, the termites can still consume the wood, they can chew it up, and they can break it into little tiny particles, but they are not able to derive any sustenance or nutrition from it. So basically, the termite can eat and it can be completely stuffed, and it can still starve to death if it doesn't have the protozoa. The other structural polysaccharide that we saw already is chitin, and chitin is found in insects and in crustaceans as part of their exoskeleton. And here we see the monomer of chitin that includes a glucose molecule, but the glucose molecule is not exactly just the glucose molecule. Over here, the hydroxyl group is substituted for an amino group that has a carbonyl, and then a methyl group. So it is a substituted carbohydrate, and polymers of chitin then form the exoskeletons of insects and crustaceans, which are together they're arthropods and spiders also. And it has a lot of applications in medicine. So we see the surgeon here, this surgical thread is made with chitin. When you have stitches that are with that thread that decomposes and that doesn't need to be taken out, that material is chitin. And chitin is also used in bandages because it has antimicrobial properties. There are a lot of other medicinal characteristics that chitin has. It's a very interesting material, and a lot of research is being done on it. In nature, chitin also provides support and protection to insects, crustaceans, and spiders. In the case of these poor crabs, it wasn't completely effective. And let's take a look finally at the fuel polysaccharides we have on the left side. We see the fuel polysaccharide in animals is the glycogen molecule. And if you look at this big molecule that is circled in the center, there is a protein, and then that is surrounded by thousands of units of glucose. Each of these tiny dots is a glucose molecule. And in plants, the fuel storage polysaccharide is called starch. And here we see an example of this starch. And here we see how the molecules form. It's a different organization. Instead of circular like you have in animal glycogen, you see a more straight lines, but each of these again is a unit of glucose. The polysaccharides, starch and glycogen, are very similar to cellulose, but the glycosidic linkages between them are different. So our enzymes are able to digest starch, but not cellulose. If you look at these molecules, these starch subunits, you see that each of the glucose monomers is oriented in the same direction. If you look at cellulose, the molecules are oriented in opposite direction. So each one is upside down with respect to the next. So that would be like in this case, if you have the first one, you have this Ch02OH on top, the next molecule would have it on the bottom, and then the next one on top again, and on the bottom, etc. We mentioned already that lipids do not form these huge polymers that we see in carbohydrates, proteins, and nucleic acids. Lipids also tend to have quite diverse shapes, so there's differences in the shape and the general structure of lipid molecules. They all have in common, though, that they are hydrophobic, so they do not dissolve in water. And some examples includes the fats, the neutral fats as they're called, waxes, which are fats that are not liquid, phospholipids, and steroids. And we're going to take a short look at each of those categories. The neutral fats are also known as triglycerides, and they consist of a molecule of glycerol, and glycerol is a sugar alcohol with 3 fatty acids attached to it. And they're called fatty acids because they have a fat component, a hydrophobic component, and an acidic component. What is the part that makes a fatty acid acidic? Well, that would be the carboxyl group on the fatty acid, this part here. And when you perform this reaction, when you add a fatty acid to a glycerol molecule, so here's the glycerol, and the yellow part is the fatty acid, we are doing a dehydration reaction. These are all performed by enzymes. So a dehydration, condensation, or synthesis reaction that we have described earlier is performed to put together the fatty acid with the glycerol molecule. And then there are 2 more positions where this can happen. So another fatty acid can be added here, and a 3rd fatty acid can be added over here. And then you have a triglyceride, or a glycerol molecule that has 3 fatty acids attached to it. Fats are used in the body, in the organism, for fuel and to store energy. So fuel to be burned immediately for calories and for energy needs, and energy storage for a later time. We distinguish between saturated fats and unsaturated fats. Saturated fats have in common that they don't have any double bonds in their hydrocarbon chains. The hydrocarbon chains are the parts that make the fatty acid fat. So this part over here is the hydrocarbon part. As the name tells us, it consists of carbon atoms that are surrounded by hydrogen atoms. So a carbon atom skeleton surrounded by hydrogen atoms. And these hydrocarbons are hydrophobic. They do not dissolve in water. In saturated fats, the carbons have the maximum possible number of hydrogen atoms about them, and that also implies that there are no double bonds in that hydrocarbon chain. Every time you introduce a double bond in a hydrocarbon chain, in order to do that, you have to lose 2 hydrogen atoms. So in order to have the maximum number of hydrogen atoms, you have to have only single covalent bonds in the hydrocarbon chain. So hydrocarbon chains tend to be straight in their shape, and therefore they're able to pack very tightly together and very compactly, and on a macromolecular scale that is going to make them solid at room temperature, like this butter that we see over here. The wax from a bee is another example of a saturated fat. In this picture here, a quite impressive picture, we see the underside of a bee, so the abdomen of a bee, and you see these small little sheets, transparent sheets of wax coming out of the bee's abdomen. Then the bees take that wax and they chew it, and then they pack it together to make their hive, so to make these cells in the hive, and those cells are used both to house the eggs that the queen lays, and also to store honey for the members of the hive. So you can imagine how much it will take to make one of those cells, and let alone the whole hive. And then human beings then can use the wax. They steal that wax and they use it for candles. And now you can also see why candle wax that is made with real beeswax is a little bit more expensive than made with other components. Beef contains both saturated fat and unsaturated fat. And the fat that we see here, that white substance that is marbling the beef, tends to be mostly saturated fat. We mentioned already that the difference between saturated and unsaturated fats was the fact that in saturated fats, all of the carbons in the hydrocarbon skeleton are joined by single covalent bonds. In unsaturated fats, on the other hand, there is at least one double bond between the carbons in the hydrocarbon skeleton. And that has as an effect that you're going to have less hydrogen atoms. Remember, for each double bond, you're going to have 2 less hydrogen atoms. And that's why they're called unsaturated. They're not saturated. They do not have the maximum number of possible hydrogen atoms linked to those carbons. We see here oleic acid as an example of an unsaturated fat. Oleic acid has exactly 1 double bond in the hydrocarbon chain, and you can see it graphically because at the double bond there will be a kink in the carbon chain. So it isn't a perfectly straight chain like in saturated fats. There's going to be a bend at every position where you have a double bond. Now, sometimes when you write a structure of a fat, when you type it up, they don't always make them curved. So you have to make sure when you look if a molecule is saturated or unsaturated as a fat, just look for the double bonds, and the double bonds indicate that the fat is unsaturated. If there is only 1 double bond, like here, the molecule is a monounsaturated fat. If there are multiple double bonds, then you can have a polyunsaturated fat. We've mentioned saturated and unsaturated fatty acids, and you have probably heard in the media about trans fatty acids. So let's take a look at what these are molecularly. Every fatty acid is going to have a carboxyl group, a carbon that is double bonded to an oxygen, as is shown here, and then also connected to a hydroxyl, and then a hydrocarbon chain. So this is the hydrocarbon chain of a short fatty acid. It has a total of 6 carbon atoms, so like 1, 2, 3, 4, 5, and 6. If you look at the unsaturated version of this fatty acid, so we have introduced one double bond between carbons 3 and 4, and when you do that, here's the double bond. When you do that, the number of hydrogen atoms to which those 2 carbons can bond is 1 less for each of them. So you're going to lose 2 hydrogen atoms in that molecule. And physically, the molecule is going to bend because in nature, these double bonds are usually cis double bonds. And a cis double bond is one in which the remaining hydrogen atoms that are on these 2 neighboring carbons are at the same side of the double bond. So, and it is a cis double bond. Artificially, it is possible to make a molecule that has a trans double bond, and that's what we see here in the lower molecule. So that molecule, the trans fatty acid, is an isomer of this one. So they have the same chemical formula, but in the trans fatty acid, as you can see, the 2 hydrogen atoms are on opposite sides of the double bond. And one thing that happens when you have a trans double bond is that now that bend that we saw earlier, you see how the shape of this molecule, it has a big bend in it, is straightened out. So this made it possible for food manufacturers to say that they were using unsaturated fatty acids, which were known to be healthy, but at the same time they would have properties that were similar to saturated fatty acids because they were straight molecules, and so they would be solid at room temperature. This molecule here is EPA, and the full name is eicosapentaenoic acid. It is one of those fatty acids that you can buy in the health food store. It is an important component of cellular membranes. In its natural form, EPA has all of its double bonds in cis form. The molecule has 20 carbon atoms and 5 double bonds. And all of the double bonds are in cis form, so it is completely bent like this. The other 2 illustrations show the same molecule, in other words, the same number of atoms, the same formula. These are isomers of each other, but in the second picture, part of the double bonds, 2 or 3 of the double bonds, are now trans double bonds. So you can see the molecule is gradually straightening out. And then finally, at the bottom, all of the double bonds are trans double bonds. So you have made the molecule from one that is completely bent to one that is completely straight. And yet, it is still a polyunsaturated fatty acid. In this slide, we're trying to make a connection between the number of double bonds in a fatty acid, the number of hydrogen atoms that it has, and its melting temperature. So we're comparing three fatty acids that all have the same number of carbon atoms. So you have stearic acid, oleic acid, and linoleic acid. Stearic acid is a saturated fatty acid that doesn't have any double bonds at all. So 18 carbon atoms with 0 double bonds. Oleic acid is an unsaturated fatty acid, but it has 1 double bond. So it is a monounsaturated fatty acid. And then linoleic acid is also an unsaturated fatty acid, but it's not monounsaturated because it has 2 double bonds. But like the other 2 molecules, linoleic acid has 18 carbon atoms. So now the question is, if you have 3 melting temperatures that are shown over here, 3 degrees Celsius, 70 degrees Celsius, and 13 degrees Celsius, which one belongs to which? So which of these fatty acids will have the highest melting temperature? Which one will have the lowest temperature? Think about it for a minute, and then when you know the answer, go to the next slide. Here's the answer to that question, along with some pictures to illustrate what we are talking about. Let's take a look first at stearic acid. Stearic acid with 18 carbons but no double bonds has a melting temperature of 70 degrees Celsius, so it is solid at room temperature. If you count the number of hydrogen atoms on the hydrocarbon portion of the chain, you will find it to be 35 hydrogen atoms. Notice that there isn't any double bonds, there aren't any double bonds between the carbons in that hydrocarbon skeleton. Then oleic acid. Oleic acid is a monounsaturated fatty acid, also 18 carbons but 1 double bond. Its melting temperature is a lot lower, 13 degrees Celsius. So that would make it a liquid at room temperature. Oleic acid is a large component of olive oil, and it has only 33 hydrogen atoms in the hydrocarbon portion of the molecule, and look at how it's bent. So it is this bending that causes the molecule to be liquid at room temperature. The idea is that if you have lots of molecules like this, they can't pack together efficiently, and so they will remain liquid at room temperature because the molecules are not that close together. And then finally we have linoleic acid. Linoleic acid, 18 carbons still, but now 2 double bonds, and its melting temperature is all the way down to 3 degrees Celsius, and it has 31 hydrogen atoms in the hydrocarbon portion of the molecule, and you see here the 2 double bonds over here. And notice also how it is bent and kinked at the places where the hydrogen atoms, where the double bonds are located. A few common oils: oleic acid, as we would see in olive oil, and then linoleic acid in grapeseed oil. That one isn't as common as the olive oil. Here are grape seeds, so they grind those grape seeds and then they can extract the oil from them. Grapeseed oil tends to be a lot more expensive than say olive oil. And then here on this one, in this part, we see stearic acid. And as you can see, stearic acid is solid at room temperature, and in purified form it can be used to make soap and also candles. So those candles would be a little bit less expensive than the ones made with beeswax. The phospholipids are another category of the lipids, and they also have a glycerol molecule like the neutral fats, but instead of 3 fatty acids attached to the glycerol in the case of the phospholipids, we have 2 fatty acids attached to the glycerol molecule and one polar group that has a phosphate functional group. So if we take a look here at the illustration of the phospholipid, the glycerol molecule is shown here. Sorry. And then the 2 fatty acids are attached to the glycerol molecule here and here. And then here on this one, on this side, is the polar group with the phosphate functional group. Here is the phosphate group. I'm circling it. If you look at a 3D representation of the molecule, it looks like this. Often in phospholipids, you'll see that one of the fatty acids that is attached to the phospholipid is a saturated fatty acid. In other words, the connection between the carbon atoms in the skeleton is single bonds, and it gives you a straight molecule. And then the other fatty acid tends to be an unsaturated fatty acid with one or more double bonds. And you can have fatty acids with lots of double bonds, like EPA and DHA have 5 and 6 double bonds respectively. So the components then, again, you have a polar head and 2 nonpolar tails, which are the fatty acid tails. And phospholipids are very important components of all cellular membranes. Phospholipids have the very interesting property of being amphipathic. That means that they are simultaneously hydrophilic and hydrophobic. The hydrophilic part of a phospholipid is the head that has the phosphate group in it, and then the hydrophobic part consists of the 2 fatty acid tails. Because of these contrasting properties in the same molecule, when you put phospholipids in water, they tend to naturally aggregate into a bilayer structure that we can see on the illustration. And they arrange themselves in such a way that the hydrophobic parts of the phospholipid, the fatty acid tails, will be facing each other on the interior of the bilayer, while the hydrophilic heads will be facing both to the inside and to the outside of the cell, for example. As we said in the previous slide, phospholipids are very important components of cellular membranes, both interior membranes and the plasma membrane. This illustration shows us a representation of a membrane, like a cross section through a membrane, and also the interaction of this membrane with the water that surrounds it. So what the people who made this illustration did is that they labeled the oxygens in water molecules, the oxygen atoms are labeled in bright green. So all of these little green spheres represent water molecules. Then the free oxygen molecules are in red. So you can see oxygen that is on the outside of the bilayer, but you can also see that it is penetrating the bilayer. Some of these red spheres are on the inside of the bilayer. We can also see the hydrophilic part of the phospholipids. They labeled phosphorus. So remember that I told you that the hydrophilic part of the phospholipid has a phosphate group, and of course there is phosphorus in the phosphate group. So all these phosphorus atoms are in the big orange spheres. And you can even see the oxygens that are associated with the phosphate group, the little red spheres that are together with the big orange ones. And anyway, it gives you an idea of how the phospholipids interact with each other. You see these little squiggly molecules are the fatty acid tails of the individual phospholipid molecules, and how they are facing each other in the middle of the bilayer. And then you see the hydrophilic heads are here, in both cases in contact with the water molecules. The steroids constitute the last category of the lipids, and steroids are all based on the cholesterol molecule, and we see the structure of cholesterol on the slide, and you can see immediately that cholesterol has a quite different structure from the neutral fats and the phospholipids. Cholesterol has these 4 organic rings that you would not see on either of these other 2 categories. Cholesterol is a very important component of the membranes in animals, and it is also the precursor of all the steroid hormones. So we're going to take a brief look in the next slide at some of these steroid hormones. In the previous slide, we commented on the structure of cholesterol and how the cholesterol molecule is the precursor for all of the steroid hormones. On this slide, I just circled the structure of cholesterol for you. So that's the cholesterol molecule. Let's take a look. I'm just going to point out a couple of steroid hormones that you may have heard of. Here is progesterone. Here is testosterone. Over here we have estradiol. And here we have cortisol. So those are just a couple of examples of various steroid hormones that have different functions in the body. Testosterone is the main male hormone. Estradiol is the main female hormone. And cortisol is used in inflammation. So you have all of these different functions for molecules that are all based on the structure of cholesterol. Here is another slide that shows us the basic similarity between hormones that are derived from cholesterol, and it also shows us what a difference a small molecule like a functional group can make. So we see that the differences between testosterone, which is responsible for many of the secondary male characteristics, such as you can see on the mane of the male lion, and estradiol, which is responsible for female characteristics. You can see here there is a hydroxyl group in the estradiol, while there is a carbonyl group in the testosterone, and then testosterone also has a methyl group over here, and that's basically all the difference between those two hormones that have very different effects on the bodies that they are working in. Another two examples of molecules that have cholesterol as their building block are cortisol, which is also known as hydrocortisone, and cholecalciferol, which is none other than vitamin D3. You can readily recognize the cholesterol structure here in the hydrocortisone. In the vitamin D3, you can see that it has undergone a few modifications, but still it is based on the cholesterol molecule. So why does cholesterol have such a bad reputation? And you're always hearing people say, don't eat too much cholesterol. Well, it is because you can sometimes find plaque deposits inside our arteries, and when you look at the components of that plaque, there is a lot of cholesterol in it. However, we have just seen in the previous couple of slides that cholesterol is synthesized in our own bodies, in our own cells. So most of the cholesterol that is in our bodies is actually derived from our own cells, and the dietary cholesterol that we consume doesn't have much to do with the cholesterol that ends up in the plaques. There are other things that may be responsible for stimulating synthesis of cholesterol and for forming these plaques. So give cholesterol a break. It isn't as bad as its reputation seems to imply. The proteins constitute the third group of macromolecules that we'll be looking at this chapter. We're looking here at three different ways of representing the protein lysozyme. In part A, we see what is known as a ribbon model, and ribbon models are particularly good for observing secondary structures like alpha helices and beta pleated sheets. So what I just circled is an alpha helix, and this part here is a beta pleated sheet. Beta pleated sheets look like flat ribbons. So the ribbon model shows this very well. Then we come to part B, which has the space filling model. And in the space filling model, you get a better idea of what the three-dimensional shape of the protein is going to be. So we see that here in the groove, you see where the substrate, in this case it's an enzyme, where the substrate would fit into the enzyme. And then finally, we have a wireframe model in C. And the wireframe model gives you the same structure as you see in the ribbon model, but you've got the functional groups of the side chains of the amino acids extending from the backbone of the protein. So you can see them here, little what looks like little wires sticking out from there are functional groups. At each corner you have a carbon atom, and each of these ends would be a hydrogen atom. Proteins perform a great variety of functions in the cell and in an organism. Here we have a list of these functions in general. So they can be enzymes, and enzymes perform chemical reactions. Trypsin is an example of that. They can be messengers in a cell or in a body. Insulin and human growth hormone are examples of protein hormones. Proteins can fulfill a structural function like keratin and collagen. They can be motor proteins like actin or myosin, which help the cell to perform certain movements or help an organism organize cells to perform mechanical motion for the organism. They can be storage proteins like casein. In the case here we see the milk on the picture contains casein. They can be defensive, so they have a defensive function such as antibodies. They can be transport proteins. Hemoglobin transports oxygen and carbon dioxide through the bloodstream. Or they can be receptor proteins, which are usually embedded in the cellular membrane, and ion channel receptors are examples of that. The building blocks of proteins are amino acids, and the connections between individual amino acids is made through dehydration synthesis, in which a molecule of water is extracted from between the two units, and then they are connected through a peptide bond. And the peptide bond is a strong, stable, covalent chemical bond. Here we see this process of making a bigger polypeptide by adding two, here is a dipeptide, and adding a monopeptide or a single peptide to that through a peptide bond. And now we have, as a result, a tripeptide. And you see here is the backbone of that tripeptide. And here are the side chains of each of the participating amino acids. The basic structure of an amino acid is shown in this slide. All free amino acids have an alpha carbon, which is this carbon that we see here in the middle. And the alpha carbon is surrounded by 4 different groups. There is always an amino group. The amino group can be NH2 or NH3+, and the amino group gives the amino acid a basic character. And there is also always a carboxyl group. The carboxyl group can be COOH, as we see here, or COO-. And the carboxyl group makes the amino acid acidic. So now you see the amino acid is simultaneously basic and acidic. Then there is also a hydrogen atom. And finally, there is an R group, or a side chain. And that R group can be many different functional groups. The R group is what makes each amino acid unique and different from the other amino acids. What is the difference between polypeptides and proteins? A polypeptide, that word refers to a string of amino acids. So lots of amino acids connected to each other through peptide bonds. When we refer to a protein, sometimes it can also be composed of one polypeptide, or sometimes it is composed of more than one polypeptide. But the polypeptides are in a very specific conformation. In other words, they are folded in a very specific way that allows them to function, to perform the function of that protein. So when you have a protein and you want to study what size the protein is, for example, then you would denature the protein. You would stretch it out until all that's left of it is its primary structure, until it's just a polypeptide. And then you can run that polypeptide on a gel, and you can compare that with other polypeptides, and you can see how many amino acids are in the polypeptide, for example, by its size. But the activity of the protein will only be there if the protein is folded into its active conformation. So the polypeptide is just the primary structure, the connected amino acids, while the protein refers to the complete structure, the folded polypeptide. And in the case of a protein that consists of more than one polypeptide, all of the polypeptides interacting with each other. We can distinguish 4 levels of protein structure: primary, secondary, tertiary, and quaternary. We're going to take a look at each of them in the slides coming up. The primary structure of proteins is then determined by the order, the sequence of the individual amino acids that compose the polypeptide chain. It is maintained by peptide bonds, and a peptide bond is a covalent chemical bond between the different amino acids in the polypeptide chain. Primary protein structure is determined by the instructions in the DNA. The DNA is transcribed to messenger RNA, and the messenger RNA is translated to protein. So the order of the amino acids is determined by the order of nucleotides in DNA and in RNA. Each polypeptide has an amino end and a carboxyl end. If we circle them, the amino end is over here, and it has the first amino acid that is added to the polypeptide chain, and that first amino acid is going to have a free amino group. The last amino acid is going to have a free carboxyl group, and that makes up the carboxyl end of the polypeptide chain. All of the other carboxyl groups and amino groups that were present in the free amino acids reacted with each other to make peptide bonds and to connect the amino acids to each other. So in the complete polypeptide chain, there is only one free amino group in the backbone and one free carboxyl group in the backbone. Now the R groups of individual amino acids may have carboxyl groups or amino groups, but we're referring here to the amino and carboxyl in the backbone of the protein. Secondary protein structure is superimposed upon the primary structure, and it consists of local areas that are either coiled or folded into specific shapes. It does not comprise the whole protein, but different areas of the protein and it can be repeated many times. So these two different structures are the alpha helix that we see here on top and the beta pleated sheet we see on the bottom. Both the alpha helix and the beta pleated sheet shapes are maintained by hydrogen bonds, and these hydrogen bonds are between non-consecutive amino acids in the polypeptide backbone. Here is an example of a protein that has lots of beta pleated sheets as part of its structure. It is called the green fluorescent protein, and it's found in nature in a jellyfish, and that some of the cells of the jellyfish are able to produce a fluorescent green color because of the presence of this particular protein. Another example of a protein with beta pleated sheets is spider silk. So we see here a spider sitting in its web, and here we see the silk coming out of the spider's spinneret at the end of its abdomen. Here we see an example of a cloth that was actually woven using silk from spiders. This particular piece of cloth is 11 feet long, and it uses a spider called the golden orb weaver, and it took the silk of 1 million spiders to make this piece of cloth. The color, this yellow color, is actually the natural color of the silk as it comes from the spider, so it's not dyed. As you can see here, the spiders themselves are quite large. There are 2 of them, and on the right side you can see the thread as it's being woven to make the cloth. So it takes the silk of a lot of spiders to make one strand of thread that can be used to weave a piece of fabric. And this is an example of a protein that has a beta pleated sheet structure. Just like secondary protein structure is superimposed upon primary protein structure, we're going to see now that tertiary protein structure is superimposed on that. In tertiary protein structure, we're looking at the folding of the complete polypeptide chain. Rather than just local areas in the chain, we're going to see how the complete chain folds in a very specific shape, giving the protein its active conformation. And you will have interactions between amino acids, and this time the interactions are between the side chains of the amino acids. In other words, between the R groups, not between the backbone parts of the amino acids. And these interactions can be any sort of bonds. So you can have hydrophobic interactions, as is shown over here. In hydrophobic interactions, what you have are water-insoluble, non-polar molecules that are making contact with each other and that protect each other from being in contact with the surrounding water. And you also have van der Waals forces that are usually found together with the hydrophobic interactions. In addition to that, you can have ionic bonds, here is an example of that. So here is a carboxyl group here forming an ionic bond with an amino group. Then we've got weak hydrogen bonds. And finally, we have covalent disulfide bridges. Each of these is going to be individual to the specific protein, and the interactions are supposed to occur in a specific place. So every time they happen in the same protein, they're going to happen in the same position, and this is what is going to give that protein its unique globular shape. Let's take a look at tertiary structure in myoglobin. Myoglobin is a protein found in muscle tissue, and its job is to bind oxygen and carbon dioxide and help exchange these gases in muscle. It consists of one single polypeptide chain, and we can see in the illustration that its secondary structure includes quite a few alpha helices. The tertiary structure then shows how these alpha helices bind with respect to each other and form a specific active conformation that allows the protein to function. Some proteins have a level of structure that goes beyond tertiary. Some proteins have quaternary structure. In quaternary structure, we see the interaction between at least two polypeptide chains in a single protein. So a protein with quaternary structure has two or more polypeptide chains that interact with each other. And the classical example that we usually see in textbooks is that of hemoglobin. We see here an example. We see the picture of hemoglobin, and it has 4 polypeptide chains that are each folded in a very specific way. There are 2 identical beta chains and 2 identical alpha chains. They fold in a specific way. Each of them has a heme unit attached to it, and that is the part that can attract oxygen and carbon dioxide. Similar to myoglobin, hemoglobin also can bind oxygen and carbon dioxide, but hemoglobin travels in the bloodstream as it is located inside of red blood cells. Every red blood cell has about 250 million hemoglobin protein units in it. Another example of a protein with quaternary structure is collagen. Now, collagen is a very differently shaped protein, as you can see here. Collagen consists of 3 chains that are mostly alpha helical, and these 3 chains are interacting with each other almost like braided with each other, and that allows it to fulfill its function as a support protein in our tissues and around our tissues. The composition of the different amino acids in a protein is very important to allow the protein to attain its normal structure and to fulfill its functions. In this example, we're going to take a look at what happens sometimes when we have mutations in a protein. And again, the example is going to be with hemoglobin. We're looking at the beta globin chain, just the first couple of amino acids. In the normal hemoglobin, the primary structure is shown over here. As you can see, the first 7 amino acids. Compare that with the primary structure of the mutated form of beta globin, the one that is found in the most common form of sickle cell anemia. Notice that there is a change in amino acid number 6. In the normal version, amino acid number 6 is glutamic acid. In the mutated version, it is valine. So that is the only change in 146 amino acids in the total polypeptide chain. What is this going to do? How is it going to affect the function of the protein? Well, let's go now to the secondary and tertiary structure. When the normal polypeptide folds, it has a shape that is a globular shape, sort of a roundish shape. But in the sickle cell polypeptide, in the beta globin protein, it has a little notch in that shape. So there is a part where there is a small a part where it is concave. Now you remember that hemoglobin has quaternary structure. You actually have 4 polypeptide chains interacting with each other. And beyond that, in normal hemoglobin, the individual hemoglobin proteins don't interact with each other. They can pass very near each other, they can be in contact with each other inside the red blood cell, but they don't stick to each other. We see that in the sickle cell hemoglobin, because of that notch in the beta globin, the proteins, the individual proteins with the 4 polypeptide chains, tend to be attracted to each other, and they form a structure that is never seen in the normal hemoglobin. They actually form a long fiber of thousands, millions of units of hemoglobin that are attached to each other. This has several consequences. One is that the protein is much less efficient at carrying oxygen and carbon dioxide than the regular version. And 2 is that it physically pulls the red blood cell out of its normal shape. This is what causes the sickle cell shape in persons affected with sickle cell anemia. The crystallizing hemoglobin fibers will actually pull the red blood cell into that shape. These sickle-shaped red blood cells then get stuck inside capillaries when the oxygen concentrations are low, and they get stuck, and they can cause clogs, and they can, tissues that are beyond the clog are then deprived of oxygen, and this is what causes some of the major problems in sickle cell anemia. We saw earlier that for a protein to attain its normal function, it needs to have a very specific shape, and that shape has to be achieved in every one of the proteins as it is made. You have to have very specific chemical bonds between very particular amino acids in that polypeptide chain. To achieve those interactions, there are a series of proteins in the cell whose job it is to help newly made proteins fold correctly. And these proteins are called chaperonins. So chaperonins are proteins themselves, and their function is to help other proteins fold correctly. They basically protect the newly made proteins from making improper contacts with their environment that could cause them to fold incorrectly, so that the correct contacts are made and the protein is folded just the right way. The name chaperonin comes from older days when there were chaperones in society. In the little inset picture, you can see the meaning of the word chaperone in the picture, where you see a young lady sitting outside and getting out, accompanied by an older lady, and she is the chaperone. And what her job was to protect this young lady from possibly improper contacts with other people, and to keep her safe and make sure that she develops into a young lady of society and everything goes correctly. There's a little bit of humor in there, but you can see how the function of this particular protein has a similarity to that. It protects the newly made proteins when they are in a vulnerable state from getting improper contact, say with water or with other proteins, and getting folded in the wrong way. How do they do that? Their shape is in the form of a barrel, and the barrel has a cap. So the newly made protein is actually going to be inserted into that barrel, where the chaperonin is going to allow all the correct connections to be made. And once the protein is folded correctly, once it has achieved its final and active conformation, then it is released out of the barrel again. And usually once they have folded correctly, they stay folded okay, and there is no more danger, so they can be released into the society of the cytoplasm. The final category of macromolecules that we discuss in Chapter 5 is that of the nucleic acids. In general, nucleic acids have subunits that are called nucleotides. And there are two major classes of nucleic acids. They are DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. Our genes are made of DNA. RNA is a molecule very closely related to DNA, and there are three major types of RNA. There are more, but there are three major types, and they are messenger RNA, abbreviated mRNA, ribosomal RNA, or rRNA, and transfer RNA, or tRNA. Two very important functions of nucleic acids are: one, to contain the information that determines the primary structure of proteins. The genes that we have that are made of DNA, the order of the nucleotides in those genes will determine the order of the amino acids in the proteins. And then another very important function is to carry energy between cellular reactions. That is usually in the form of the molecule called ATP, or adenosine triphosphate. Let's make a point-for-point comparison of the two nucleic acids, DNA and RNA. The acronym DNA stands for deoxyribonucleic acid, while RNA stands for ribonucleic acid. You can see that DNA is a modified form of RNA. It's just like RNA, but it is missing an oxygen somewhere, and we're going to see that a little bit later. The molecule of DNA in nature exists as a double-stranded molecule. RNA, on the other hand, is made as a single-stranded molecule, and some of its parts can then be double-stranded, but in general RNA is single-stranded while DNA is double-stranded. Both DNA and RNA are made inside the cellular nucleus, but DNA stays in the nucleus, so it's made in the nucleus and it's used there as well, while RNA is going to be made in the nucleus and then it's going to be transported to the cytoplasm and used in the cytoplasm. DNA contains all of our genes. You can almost compare it to a reference encyclopedia that has all the information that is necessary to make a new cell. RNA contains a working copy of one or a few genes. In other words, RNA molecules don't contain the comprehensive information that is present in DNA. RNA molecules are individual copies of small parts of individual genes in the DNA. So if DNA is the big reference encyclopedia, RNA is a few pages here and there that are copied from the encyclopedia and that are used to actually make things using the information in the RNA. The DNA molecule is extremely long. It's millions of nucleotides, billions of nucleotides. If you take it all together, the human genome is 3.2 billion nucleotides in its haploid state. RNA molecules are generally not nearly as long. An RNA molecule can be from a tRNA molecule that's about 75 nucleotides to several hundred nucleotides to thousands of nucleotides, but never millions of nucleotides as we see in DNA molecules. An individual nucleotide, both in DNA and RNA, has three parts. Remember we said that the nucleic acids have as their building blocks these units that are known as nucleotides. In each nucleotide you will find the following three parts. A phosphate group that we're circling over here, so a phosphate functional group. This is the part that makes the DNA and the RNA acids. Then there is a pentose sugar. We're circling that one over here. And the pentose sugar means that it is a sugar that has 5 carbons in its skeleton. The carbons are shown here. So number 1, 2, 3, 4, and 5. And notice they have the numbers 1' to 5'. The carbons in the sugar are always numbered with that little prime symbol. And then finally there is a nitrogenous base. Nitrogenous base, it has nitrogen atoms on it, and depending on the base there are different numbers of nitrogen atoms associated with it. We'll take a look at the specific bases in an upcoming slide. The differences between DNA and RNA in terms of their nucleotides are found both in the nitrogenous bases and in the pentose sugar part. Let's take a look. In the nitrogenous bases, in DNA we have 4 different ones, and they are cytosine, thymine, adenine, and guanine. In RNA we basically also have 4, but there is a slight switch. So in RNA we've got cytosine, uracil, adenine, and guanine. So you can see that DNA and RNA have in common the bases cytosine, adenine, and guanine. And then thymine is only found in DNA, while uracil is only found in RNA. That's as far as the nitrogenous bases are concerned. Now come the pentose sugars. As we said before, DNA stands for deoxyribonucleic acid, while RNA is ribonucleic acid. Those names, the ribo and the deoxy, refer to the sugar part. So RNA is a ribose sugar, a pentose ribose sugar, and you can see the part in which is different from DNA is in this hydroxyl group at the 2' position. If we compare that with the sugar in DNA's sugar is deoxyribose, and you notice how instead of a hydroxyl group at the 2' position in DNA, there is just a hydrogen. That is why it is called deoxyribose. It's missing an oxygen. And then finally, the phosphate group is identical between the two. So both DNA and RNA have that same phosphate group. The sugars and the phosphate groups form the backbone of the DNA double helix or of the RNA single-stranded molecule. So let's just circle those. So you've got here a sugar, and that is connected to a phosphate, which is again connected to a sugar, and that is connected to a phosphate, and you can see you can go on, sugar, phosphate, sugar, phosphate, sugar, phosphate. That forms the backbone of the molecule. The connections, individual connections between one nucleotide and the next on a single strand of DNA or on a strand of RNA are phosphodiester bonds. They are covalent bonds. So each connection between a phosphate and a sugar molecule is called a phosphodiester bond. Then on the inside, in the case of the DNA molecule, you have the bases. So you've got a base here and another base there and another base here and another base there. We're only looking at one strand in this case, so it could be a single-stranded DNA molecule or just a regular RNA molecule. If you have a double-stranded DNA molecule, then the two strands connect to each other via the bases. So the bases are going to be on the interior of the molecule. Just like carbohydrates and proteins, nucleic acids form polymers, and these polymers are covalent bonds between one nucleotide and the next, and we showed that in the previous slide where you could see the connection between the phosphate group and the sugar molecule in the individual nucleotides, and that was called the phosphodiester bond. In RNA molecules, we usually have one polynucleotide chain, while DNA forms a double helix with the two molecules connected to each other through the bases, and the bases are connected with hydrogen bonds between them. They're connected in a very specific way. The adenine base bonds to the thymine base. If it is a DNA to an RNA, or if it is in an RNA part that is double-stranded, adenine will bind to uracil, and cytosine is going to bind to guanine. We take another short look at the structure of DNA. It forms a double helix as its secondary structure. Remember, the primary structure is the order of the individual nucleotides in one strand, and then the secondary structure is formed by the double helix. The double helix has a backbone of alternating phosphate groups and sugar molecules that are connected through covalent phosphodiester bonds. So there are two backbones in this molecule. There is one shown for each of the two strands. So I circled one, or I followed one in purple. Let's follow the other one in yellow. That's the other backbone for the other side of the double strand. And then the bases are connected to each other. The bases are on the interior and are the horizontal parts that we see. What color shall we give them? Let's make them white. So the bases are these parts on the interior of the molecule, and they are connected to each other using hydrogen bonds in which A is bonded to T and C is bonded to G. The other very important function that nucleic acids fulfill is that of energy carriers in cellular reactions. The great majority of our cellular reactions are powered by the nucleotide adenosine triphosphate, or ATP. Adenosine triphosphate does this by donating either one or two of its phosphate functional groups through the process of hydrolysis. We saw at the beginning of the chapter that hydrolysis means breaking with water. So water is going to be used to break the molecule of the ATP molecule and separate a phosphate group from it, or two phosphate groups, and that releases energy. In the opposite direction, when we make ATP molecules, after all, you have to make them to have them later to use them. When we make ATP molecules, we need to use ADP and then add inorganic phosphate to the ADP molecule, and that takes energy. The major cellular processes that allow the making of ATP are photosynthesis, that is performed by plants, algae, and some bacteria, and cellular respiration, that is basically performed by all cells.